Good day, this is Professor drwillphd, PhD. And today's Friday, April 22nd, 2022. It is 6:12 and 24 seconds, AM Eastern Standard Time. Today's research, mutagens. So let's hop to it here. Mutagens across animal kingdom shed new light on aging. Quantity of mutagens acquired similar over a lifetime of 16 species, despite vast differences in lifespan and body mass. Date April 13, 2022. Source: Wellcome Trust Sanger Institute. A new study compares the accumulation of mutagens across many animal species and has shed new light on decades-old questions about the role of these genetic changes in aging and cancer. Researchers found that despite huge variation in lifespan and size, different animal species and their natural life with similar numbers of genetic changes. Genetic changes, known as somatic mutations, occur in all cells throughout the life of an organism, and this is a natural process, with cells acquiring around 20 to 50 mutations per year in humans. Most of these mutations will be harmless, but some of them can start a cell on the path to cancer or impair the normal functioning of the cell. And since the 1950s, some scientists have speculated that these mutations may play a role in aging, but the difficulty of observing somatic mutations has made it challenging to study this possibility. In the last few years, technological advances have finally allowed genetic changes to be observed in normal tissues, raising hopes of answering this question. And since cancers develop from single cells, species with larger bodies, and therefore more cells, should theoretically have a much higher risk of cancer. Yet cancer incidence across animals is independent of body size. Animal species with large bodies are believed to have evolved superior mechanisms to prevent cancer. Whether one such mechanism is a reduction in accumulation of genetic changes in their tissues has remained untested. Researchers set out to test these theories by using new methods to measure somatic mutation in 16 mammalian species, covering a wide range of lifespans and body masses. And this included species such as a human, mouse, lion, giraffe, tiger, and the long-lived, highly cancer-resistant naked mole rat, with samples provided by a number of organizations, including the Zoological Society of London. Whole genome sequences were generated from 208 intestinal grips taken from 48 individuals to measure mutation rates in single intestinal stem cells. Analysis of the patterns of mutations or mutational signatures provided information on the process at work. The researchers found that somatic mutations accumulated linearly over time and that they were caused by similar mechanisms across all species, including humans, despite their very different diets and life histories. The evidence of a possible role of somatic mutations in aging was provided by the researchers, discovering that the rate of somatic mutation decreased as the lifespan of each species increased. And to find a similar pattern of genetic changes in animals as different from one another as a mouse and tiger was surprising. But the most exciting aspect of the study has to be finding that lifespan is inversely proportional to the somatic mutation rate. And this suggests that somatic mutations may play a role in aging, although alternative explanations may be possible. Over the next few years, it will be fascinating to extend these studies into even more diverse species, such as insects or plants. After accounting for lifespan, the authors found no significant association between somatic rate and body mass, indicating that other factors must be involved in larger animals' ability to reduce their cancer risk relative to their size. The fact that differences in somatic mutation rate seem to be explained by differences in lifespan rather than body size suggests as although adjusting the mutation rate sounds like an eloquent way of controlling the incidence of cancer across species, evolution has not actually chosen this path. It is quite possible that every time a species evolves in larger size than its ancestors, as in giraffes, elephants, and whales, evolution might come up with a different solution to this problem. We will need to study these species in greater detail to find out. Despite vast differences in lifespan and body mass between the 16 species studied, the quantity of somatic mutations acquired over each animal's lifetime was relatively similar. On average, a giraffe is 40,000 times bigger than a mouse, and a human lives 30 times longer. But the difference in number of somatic mutations per cell at the end of a lifespan between the three species only varied by about a factor of 3. Animals often live much longer in zoos than they do in the wild, so our vets' time is often spent dealing with conditions related to old age. The genetic changes identified in this study suggest that diseases of old age will be similar across a wide range of mammals. Whether old age begins at 7 months or 70 years, it will help us keep these animals happy and healthy in their later years. Understanding the exact causes of aging remains an unsolved question and an area of active investigation. Aging is likely to be caused by accumulation of multiple types of damage to our cells and tissue throughout life, including somatic mutations, protein aggregation, and epigenetic changes, among others. Comparing the rates of these processes across species with very different lifespans can shed light on their role in aging. Aging is a complex process, the result of multiple forms of molecular damage in our cells and tissues. Those somatic mutations have been speculated to contribute to aging since the 1950s, but studying them has remained difficult. With the recent advances in DNA sequencing technologies, we can finally investigate the roles that somatic mutations play in aging and in multiple diseases. That this diverse range of mammals in their lives with a similar number of mutations in their cells is an exciting and intriguing discovery. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.